Thank you everyone for joining us today. And yeah, thank you for joining and for the next instalment of our research webinar series, which is part of our Staying Connected programme. And we're thrilled to see we're joined by many of our wonderful supporters, including members of our patient research network, who I think have come to so many of these research updates, OCA trustees and our patrons too. So a special welcome to you all. As many of you know, it's been a challenging 12 months for all charities, and Ovarian Cancer Action is no exception. At the beginning of the pandemic, the outlook for fundraising looked pretty bleak and very uncertain. It has indeed been a challenging year for everyone, including us, but we're very pleased to say that despite this, our team have achieved some fantastic things this year, including launching the Staying Connected programme to support patients through the pandemic, received a large grant from the government's tampon tax to address health inequalities for women with ovarian cancer, and now thanks to you, our wonderful supporters, we've been able to fundraise enough money to get our research back on track much quicker than we anticipated. The project we're going to be hearing about tonight was one of the 3 new research projects that were delayed by the pandemic. The support given by many of you already means that we can get this work underway now to ensure that patients can benefit from research breakthroughs as quickly as possible. This evening, Professor Ahmed Ahmed will be talking about his research to find new, kinder and more effective treatments for women with ovarian cancer. What Professor Ahmed probably won't tell you is that this project was the winner of our 2019 Immunotherapy Challenge, where we asked scientists from all over the world to submit proposals that would unlock the benefits of immunotherapy that we've seen in other cancers. Our expert scientific advisory board judged this project above all others to have the most potential to make effective immunotherapy treatment a reality for ovarian cancer patients. We couldn't be more excited about what the future holds for this research and its potential to impact patients' lives. Professor Ahmed is Professor of Gynecological Oncology at the Nuffield Department of Women's and Reproductive Health at the University of Oxford. And his main research interests focus on the surgical, medical and fundamental research into ovarian cancer. And he leads a fantastic team of OCA-funded scientists in his lab who are making huge strides in the early detection and treatment of ovarian cancer. He's also a gynae-oncology surgeon in Oxford, so a very busy man. Thank you, Ahmed, for taking the time from your busy schedule to talk to our supporters this evening. I know this is the second webinar you've hosted for us very recently, and we'll let you take a break after this. But before I hand over to Ahmed, please can I ask that you ask any questions through our Q&A function, and we can get Ahmed to answer those for you after the session. We also have had some questions in beforehand, so thank you to those who sent those in, and I'll ask those to Ahmed as well. We kindly ask that you do not ask any personal medical questions, as Ahmed won't be able to answer those. So without any further ado, thanks again for joining us, and I'll now hand over to Ahmed.
Thank you very much, Faye, for the introduction, and thank you for the audience of this webinar. And just before I start, I want to say a big thank you to the charity, and I really take my hat off to you for all the great work that you have done, and for continuing in such difficult, very, very difficult circumstances of the pandemic, how you managed to keep going and to fund our research is really commendable. So thank you for persevering and for supporting us. So today I'm just going to talk about our research plans for harnessing the power of the immune system to fight ovarian cancer. And just as a way of introduction, I'm just going to talk to you about the key concepts. So the key concepts is that in tumors, there are alterations in the DNA that occur, and we call these alterations mutations. And these mutations make the proteins appear foreign to our immune system. And that's how our immune system starts to react. And it reacts by generating antibodies that circulate in our bloodstream, but also reacts by through cells, predominantly the subset of cells called T cells. So I'm going to explain these concepts in more detail, but that's in broad terms. What we're trying to do is to essentially harness this power of reaction of our immune system to mutations, and essentially so that we can overpower the cancer cells and promote this immune response and to fight cancer. So just start by explaining some basic concepts about our DNA in our body. So in each cell, there is a DNA as a nucleus that is really composed of the basic components of 4 nucleotides. They're really summarized in letters, A, C, G, and T, as shown here in these colors. I'm going to talk about that in a bit of a detail later on. But there are about 3 billion of those letters, and these sort of letters make the code of the DNA. And it's in that DNA, encrypted within those letters, are the sequence of the DNA are essentially the message of the RNA and the proteins in the cell, as I'll explain. So Watson and Crick discovered the DNA double helix, the structure, and got a Nobel Prize for it. But it's really this genius, Fred Sanger, who discovered how to read the code of the DNA, and also not just the DNA, but also the protein code, and got the Nobel Prize twice. And it's thanks to him that we're now using the basic concepts of how to use the DNA in order to discover not only the basics of the DNA sequence in our bodies, but also the alterations that are, as I mentioned, what we call mutations. Some of the key basic concepts I'm going to lay out to you at the beginning. So as I mentioned, in every cell there is this DNA code, and it's composed of those 4 letters, A, T, C, and G, and they are ordered in that particular manner. So this DNA, there's 3 billion of those letters in our genome. This DNA then gets what, as we call, transcribed to a message, and that message is called RNA. That message carries the code of proteins, and proteins are essentially the workforce in the cell, and those proteins give the cells their characteristics and their identity. Now, as you can imagine, we need different cells in the body make different functions. So there is therefore certain proteins in different types of the body. So for instance, the type of proteins that we will find in the skin, abundant in the skin, will be very different from those in the liver or those in the small bowel, for example, because they do different functions. So it's that protein abundance in particular cells that give the cell its identity and type. Okay, so the basic concept is really outlined in this diagram, as I said to you, that in cancer you get mutations, and the mutations make those proteins appear foreign to our body. So if you imagine that if you're attacked by a bacteria or a virus, for instance, we're all very aware now of viruses, and the proteins on these bacteria or viruses would generate an immune response. In cancer, the concepts are very similar. So those sort of cancer cells here in blue would have these mutations that are presented on the surface of the cell. And then you have essentially T cells that are reactive to those mutations on the surface of the cell. And the idea of cell therapy, which is really going to be the main focus of our research and the main focus of this talk, is to find those purple cells that target those mutations on the surface of the tumor and expand those cells in order to deliver them back to the patient to fight tumor cells. So what's the dream? Well, the dream is really outlined here in this figure. So this is really talk is timely because it coincides with a publication that we have in the British Journal of Cancer that is in press. So this is not even the press. I'm giving you a sneak peek of what we are going to be publishing in the next week or two. And the first author of this is Dr. Matteo Morotti, who was a member of my lab before going to Switzerland to start his senior career in gynecological oncology. And the dream, as we described, was a group of us met last year before the pandemic. So these were clinicians, scientists, cell therapists, patients and patient advocates to discuss this dream of cell therapy. And this publication that we have in the pipeline essentially summarizes the thought process and the discussions that we had in that symposium last year. The idea is really that if you have a tumor, that tumor you will contain tumor cells and T cells. You get the tumor and you do a process called sequencing, which is essentially what I explained to you, a modified version of Fred Sanger's method that enables us now to generate the genetic code from a patient in about a week, as opposed to 10 years, which is the time it took us to generate the first genetic code of humans. But the idea is that you sequence the tumor, you identify those foreign proteins, which we call antigens or neoantigens, and identify those T cells that react to those neoantigens, expand them, and then give them back to patients. There are many types of these foreign antigens, or these neoantigens. There are ones that result from a change of a single letter. So that one change of one letter can be translated to a different protein. There are also things that are results from a nonsense, like a stop of the message, of the expression of the message, or the abundance of the message. There are others that result from deleting one or two or more letters. And also there are others that result in actually after the protein being present in the cell, the protein can be modified, and we call these post-translational modifications. Not all of these potential antigens or foreign antigens can stimulate our immune system in the same way, but there is an abundance of those different types of altered proteins that one can use in order to find out reacting T cells, or could use them themselves as potential vaccines to stimulate the immune response in patients with cancer. We really, really live in an exciting time, because we now have many technologies that enables us to get a tumor, and then by a variety of what we call omics techniques, DNA and RNA sequencing, looking at protein sequence, even looking at sequencing of individual cells, cell by cell within a tumor, to get the entire genetic code, sequencing T cells to find out the exact sequence of the T cell receptor that is the key component that essentially fights the foreign protein on the cancer cells. By doing a variety of those sequence methods, one can really get a very deep understanding of a tumor that would enable then selecting the best possible vaccines or cell therapies. In our proposed research, we'll be using really pretty much all of those techniques to characterize tumors. But there are really major obstacles and difficulties in tumors that to realize the dream. And those starts from cancer cells themselves to the environment in which these cancer cells survive. So let me explain to you. As the tumor develops, it develops from a normal cell. And then you get a variety of those mutations, and you get then the cell transform and becomes bigger. And then as it evolves, it would essentially accumulate more mutations along with time. Now, this process from a normal cell to actually a tumor presenting in a patient can take, in ovarian cancer, 10 to 20 years. But at the end, what you will have what you are left with is a tumor that have different types of cells, and each of those types will have certain characteristic mutations, their own set of mutations. So if we were to develop a vaccine that target particular mutations, then yes, you may end up fighting only a proportion of the tumor, but not all the tumors. So that's one thing really to consider when we are designing cell immune therapies or vaccines to fight tumors. There is also a great variety of suppressive factors that surround the tumor. Please don't worry about those names, but they are really, I'm putting this slide to show you that there are so many ways by which the environment of the tumor and by which the tumor itself can produce factors that can dampen the immune response, can weaken the immune response of our body, and therefore weaken the ability of T cells to tackle cancer cells. And what you end up with is T cells that suffer from, as the name really implies, exhaustion. And these T cells are unable to fight the cancer, to fight back the tumor. And some of these factors that dampen the immune response and slow down and exhaust these T cells, some of these factors have therapeutics that are actually already in the clinic. One of them, for example, is an antibody that blocks this PD-L1 receptor. And by blocking this factor called PD-L1, you're essentially rejuvenating these T cells and removing the brake from these T cells. And therefore, these T cells become more effective in killing cancer cells. Now, this particular drug, the anti-PD-L1 and also the anti-PD-1, is currently in the clinic and is showing major success in treating certain types of cancers like melanoma. But unfortunately, its use in ovarian cancer has not really shown a great promise to date, which means that we need to think outside outside this limited factors of PD-1 and PD-L1 in order to find potential immune therapeutic options. The good news is that we can take those T cells and condition them outside the body to rejuvenate them, to give them power, and then put them back to patients to fight cancer cells. And this rejuvenation process could be as simple as culturing them to expand outside the body, but also could include things like engineering these T cells genetically or by using protein engineering in order to generate T cells that have favorable characteristics that are capable of killing cancer cells than non-genetically engineered native T cells. Okay, but the process that I described to you, while very, very exciting, the prospect of generating cell therapies for patients is very exciting. It is actually faced with many, many challenges that I'm going to describe to you. The differences between a cell therapeutic, the next generation cell therapeutic, shall we say, compared to a pill or an injection that we would traditionally use to treat cancer patients. The difference is that you need to have these cells taken into a highly specialized place that is called a cell manufacturing facility. And those cell manufacturing facilities would have clean rooms that prevent these cells from getting contaminated by infectious agents like bacteria or viruses. And this is like really fine art. We need specialists, so we call those manufacturing specialists, that can handle these cells and expand them. So it's not only finding the correct protocol for expanding the cells, but actually having specialists and facilities that are capable of taking those cells and expanding them and changing them to become a therapeutic that can then fight cancer back in the patient. The closest to this analogy would be the bone marrow transplantation. This is really the closest thing to what I'm describing to you about cell therapy. So you have the patient here in the hospital. You need to take those cells from the patient to a manufacturing facility to be processed. Now, these manufacturing facilities, there are not many of them around the country or around the world. But what you want to be able to do is to transport them to backwards and forwards from these manufacturing facilities. This is some really important logistical consideration about how you keep those cells alive and well during that journey back and to and from the manufacturing facility. You also have a lot of issues about governance and administration, and also receipt and anonymization, in order to make sure that any patient identifiers are protected and governed appropriately. You need to keep an inventory of your cell therapies, as I mentioned, transport. But this also raises some very, very important practical considerations about regulatory issues and reimbursement issues. Let me just talk to you a little bit more about that. So how would you regulate a therapeutic traditionally is very different from how would you regulate a cell therapy product. So traditionally, if there's to regulate a pill, and by regulate, I mean find out that it's effective, both effective and safe for the patient. So you do that in batches. You generate a batch of off-the-shelf therapeutic that can last for years from a pharmaceutical company. You put that, you distribute it to pharmacies, and that every batch is tested for its efficacy and for its safety and for its standards. Imagine that you have to do that process for every patient individualized. You want to essentially quality control every patient if we're talking about individualized cell therapy, or for every group of patients if we're talking about personalized, i.e. developing cell therapies that are personalized for certain types of cancers. So that's the regulatory issue that is there, and the regulatory challenge. There's also reimbursement challenge. The reimbursement here is important because if you think about it, when you're generating these cell therapies, the current existing cell therapies that exist currently for treating some forms of blood hematological malignancies or blood cancers, they cost about £400,000 plus or minus. That's at least that's the advertised cost, and £400,000 per patient. Now, that's very, very, very expensive. However, those therapies in those indications are curative. So therefore, they are considered very, very cost effective because they can produce a cure for patients, and then patients don't have to go through rounds and rounds of treatment. So when you are developing a cell therapeutic option, one needs to take into account also these practical issues. And then finally, the reality is, while we as doctors are very much used to pill therapies, very much used to looking after patients for administering pills or injections, we're not really very well trained for administering cell therapies. So these are like really major initiatives around the country and around the world to train doctors for to administering cell therapies, for looking after patients who are going to be receiving cell therapies. So these are all challenges, but they create excitement, because if you are able to overcome and solve these issues, then, and if you're able to develop the right therapeutic, as I mentioned, the examples from blood cancers is that you can produce cure. So T-cell therapy is very promising, but highly challenging in ovarian cancer. Why? So as I mentioned, there is cell therapies have produced cure in certain types of blood cancer, but hasn't been that successful in solid tumors, in non-blood cancers, with few exceptions, like again in melanoma. And we believe that one of the reasons for that is that a tumor like ovarian cancer does not have a lot of these single nucleotide variants or mutations compared to melanomas. So therefore, it makes it very important that when you are looking from your limited number of mutations that you find in ovarian cancer, you are really super selective on selecting the ones that are most likely to succeed as a vaccine or as a guide for cell therapy. The other point is that if you think about these T cells as soldiers, well, you want the soldier to come to confront its enemy. And that's easy in a blood because those cells are in very close contact. These T cells are in very close contact with circulating cancer cells, either in the blood or in the bone marrow. In solid tumor, those soldiers need to find the tumor. You inject the T cells back to the patient, but they need to find that tumor somewhere. And also not just that they need to find it and then go and persist in the vicinity of the tumor in order to fight the tumor back. So finding those subsets of T cells that can do that is so important. So this really summarizes our research project. So our research project is really dealing with these two major issues. One is which foreign protein, which antigen that one can select for developing cell therapy. As I mentioned to you, when you are dealing with cancers that have many foreign antigens, many mutations, that's less of a problem. If you're dealing with an ovarian cancer, it is a major problem. So you really have to be very mindful about which mutations you select, and also look at all different types of alterations. And the second point is, the second key question in our research is, which T cell type can be most effective for cell therapy? When I describe to you there are T cells, there are subtypes of those T cells, and those subtypes are different in their function and their ability to fight back the tumor. So this is coming back to this slide. I mentioned to you that there are many different types of mutations and alterations, and they exist in different places within the cell, inside the cell. And a key challenge is to identify the most effective, the most effective mutations, the mutations that are most capable of mounting an immune response in the patient. We have clues. This is a paper that we published last year, showing, for example, that mutated proteins that reside in an organelle called the mitochondria are much better at inducing an immune response in patients compared to other forms of mutations. So we want to really continue along that line of research and try to identify other subsets of mutations that can that could be favorable mutations to go after. The second point is which type of T cells. As I mentioned to you, not all T cells are the same, and there are different subtypes. And our research that was published last year gave us again a clue to suggest that there are certain subtypes of T cells that already reside within tissues In this case, in this paper, we describe a subtype of T cells that reside within the fallopian tube itself. They have certain favorable characteristics. We want to study those and see if those characteristics can be harnessed and taken forward for cell therapy for patients with ovarian cancer. So finally, I want to say that we are very close to getting started. We are recruiting our postdoc to start on this exciting project, thanks to the funding that we are receiving from Ovarian Cancer Action. Thank you all for listening, and I'm happy to take any questions.
Thank you, Ahmed. That was a fascinating insight into your new research, and I really appreciated the breakdown of how the 40 proteins are produced by mutations in our DNA and linking that through to how they can be used as targets for the re-energized T cells, and also of your explanation of why it's more difficult to find those targets for ovarian cancer as well. And it's also interesting how the tumours have that almost mosaic composition of different types of mutations. And I thought the diagram you showed, showing the timeline of those mutations, was excellent at demonstrating that. And finally, I really appreciate the context you gave around what would be required to provide cell therapies to patients. I'm sure the viewers appreciated that too. And hopefully over the next years, we'll have the infrastructure and resources like you described to provide life-changing treatments like this. So thank you, Ahmed.
Thank you, Faye. I just wanted to mention one point related to this, is that really the challenge that Ovarian Cancer Action posed to us as in the research community when Ovarian Cancer Action made this funding available for a competition, was really a key component of that was that it needs to be research that is patient-facing, with an idea of translatable, that want to make discoveries and fundamental understanding that can then be translated to patients. And that really is something that is key to my understanding of what this project is about. It's really, really wholeheartedly trying to find ways to accelerate that process and take it our findings to available patient therapeutics.
You're right, that's our top criteria when we're reviewing research funding applications, because we want to ensure that supporters' donations are used on, like you said, the best research that is going to be translated to patient benefit as quickly as possible. So yeah, no, that's fantastic. Thank you, Ahmed. And so we're going to go on to questions now. I'm going to start with the questions that were sent in beforehand, but this is your time to type out any that you have now. So please ask any questions that you have. So the first question was, what is the difference between a therapeutic vaccine that you've described and a preventative vaccine like the HPV vaccine for cervical cancer?
Yes, so this is a really good question. It's really in terms of manufacturing or making the vaccine, the concepts are very similar. Essentially, what we're trying to do is we're trying to find out, for example, in the case of an HPV vaccine, we know that these HPV virus can predispose to cervical cancer. So what we're doing there is say, okay, well, HPV would stimulate the immune response because it's a virus and has foreign proteins, and therefore we try to take those proteins of the virus, make a vaccine, and use that vaccine to stimulate immune response, with the idea of preventing the tumor from starting in the first place by eliminating the virus. In this case, in the case of therapy, it's different. We are using tumor antigens, tumor mutations that transcribe and translate to neoantigens, and we're trying to use those as if they were viruses, and then stimulate the immune response to get rid of existing tumors. So while the concepts are very similar, one is to really prevent a tumor forming in the first place, and the other is to try to eliminate the tumor or reduce its size.
Great, thank you. And then next up we have, will this help with all types of ovarian cancer?
Yes, actually, I think so. Actually, if I may say, for a change, it's usually, as researchers, we focus on different individual types because that's the right way to do it, to understand the characteristics of individual types. But cancer cell therapy in general, actually, the methods that are used are pretty much agnostic to the type of ovarian cancer. So the answer is yes. So this could potentially benefit all types of ovarian cancer.
Great. And someone else has asked, would this type of therapy work on people who don't have the BRCA gene?
Yes, the answer again is yes. They should do, because again, all cancers would have genomic alterations and mutations, and therefore whether or not there is a BRCA mutation is not as crucial.
Great. And then we've had a question through that says, would being high MSI ovarian cancer be beneficial for this research?
Yes, because I think so the idea of having more mutations is great for immune therapy. The more mutations you have, as is typical for microsatellite instability or MSI, the more likely the immune therapy in general will succeed. And that includes injections like anti-PD-1 or anti-PD-L1 or cell therapies. So the answer is yes. If there were microsatellite instability containing tumors, then yes. The difficulty is that ovarian cancer is not known for having microsatellite instability, unlike uterine cancer, for instance, or unlike colon cancer. But if there was exceptional cases where there is MSI, then yes, this would help.
Great. And then this next question, it's quite a tricky question to ask because there's so many factors that go into this that can be hard to predict. But someone's asked, how soon do you think you'll get to clinical trials? Quite a big question.
Actually, I think it's a great question. It's a fair question. I don't have the answer to it, but what I can say is that it is absolutely in our mind as team of researchers that we want to be able to take these cell therapies to patients. I think the excitement here from my side and the optimism is that a cell therapy is something that you can take from that individual patient, make the cells, condition the cells, and then give back. Unlike even I described a lot of challenges in manufacturing cell therapies, but actually at its heart, it is from the patient back to the patient. And therefore, for me, it seems to me a much more predictable process compared to, for instance, if you are trying to develop a completely new therapeutic, a new chemical, because then you need to go through many, many iterations of chemical modification in order to make this the best chemical modification that can be then taken to clinic. So here, the barriers are what I described. If one can overcome those barriers, then I hope that this could be taken as quickly as possible to clinical application. If I want to put some years on it, I would hope to be within, let's say, 3 to 5 years.
And that's quite quick, as you say, in comparison to the traditional routes for treatment. So that's
yeah. Yes, maybe I'm being over optimistic, but I think I'm really, that's currently my hope, is that we would go to clinical trials as soon as possible.
No, that's a very exciting prospect. So thank you to whoever sent that question in. And then someone's asked, is there any existing infrastructure in place now to support this? So I suppose out of all the barriers that you showed, where have we made most progress in?
So I think actually we have in the UK in particular, and a few other countries. Within the UK, we have the edge because there was a government initiative that was started a few years back called the Catapult Cell and Gene Therapy, along other initiatives. But the Catapult Cell and Gene Therapy was established essentially really to take such cell therapy option forward, to propel this form of cell therapy forward, to make it a reality, not just for cancer patients, but for other indications. So I think that the infrastructure is, it is not yet a widely available, but it is certainly, there is already the infrastructure is starting to become established in many centres around the UK, thanks to initiatives like Catapult and other initiatives.
Great, thank you. And if anyone has any last minute questions, if not, we can end the session there. Well, thanks again to all of you for joining us this evening, and a massive thank you to Ahmed for taking the time again to present this webinar for us. Yeah, thank you to everyone for joining and for supporting us, and hopefully we'll see you again at a future session. So thank you and goodbye everyone. Thank you, Ahmed.